Hi, I am Pritam Saha, Department of Chemical Sciences, ISR Mohali. My thesis title is When Protein Falls Silent, Uncovering the Misfolding Roots of Hearing Loss. Currently, about 5% of total human population are suffering from hearing loss. It will reach 10% by 2050, approximated by WHO. Hearing loss is not only found in elderly people, but can also be found in children. When it is found in children, it is much more severe and called progressive hearing loss. This progressive hearing loss is happening because of mutation in tippling proteins. What are those? To understand that, let's see how we hear sound. So, sound wave goes from outer ear to middle ear to inner ear. Inside inner ear cochlea, there are these cilia-like projections forming a staircase formation. These are called stereocilia. The longer stereocilia connects with the shorter stereocilia via tipplings. These tipplings are the protein of cadherin 23 and protocadherin 15 forming a heterotetrameric handshake formation. The stippling proteins filters the force and transduces the mechanical signal into electrical signal by opening up the ion channel. Mutations in these proteins have been associated with hearing loss. One such mutation is S47P. In mice having this mutation, we have found the stereocilia are damaged. We wanted to understand how this mutation changes the force filtering ability of the tippling. That's why the damage is happening. To understand that, the only thing we can go to is single molecule force spectroscopy. However, the most recent and the current techniques like AFM does single pulling for the single event. But inside physiological condition, we are having repetitive perturbation for a long time. To mimic that, we have built a magnetic tweezer system in our lab from scratch, and we also have developed a attachment strategy at single molecule level. So what this does is basically we use magnet to trap a magnetic bead which is covalently attached to the protein of interest, which is again covalently attached to the surface. Now we can pull this protein as much time as we want, as long as we want, and in any form or fashion as we want, meaning stochastic or oscillatory or monotonous. Using this magnetic tweezer, I have given repetitive force oscillation to these proteins of mutant and wild type. We have also calculated the mechanical stability and the kinetic rates. In silico data showed the mutation causes intra-domain hydrogen bond to disrupt, meaning there are less hydrogen bonds and weaker hydrogen presence in mutant. And while giving repetitive oscillation, we have seen that the refolding rate for the mutation gets slower, which is basically having some fatigue. So we believe the mutation is causing the protein to fold slower. That is why it cannot fold back to its original conformation before the next pulse comes. And that is how, with long term, it is damaging the protein and changing the force filtering ability of the tippling. That's why the hair cells is getting damaged. The hair cells are non-regenerative in nature. That's why once they get damaged, we lose our hearing ability. For more information, you can see my paper. Thank you.